Good morning, everyone. Good afternoon, everyone. Welcome to the webinar. I'm Kabir from Battle River Research Group, and I'm thrilled to be your host for today's session. Before we dive into the insightful discussion with our special guest, Dr. Boyd Murray, on integrated pest management, let's take care of some housekeeping. If you have any questions, feel free to ask at the end of the webinar or drop them in the Q&A section or the comment section. Now, without further ado, let's give a warm welcome to our expert speaker, Dr. Boyd Murray. Welcome.
Okay, thanks very much. All right, so today I've been asked to speak about integrated insect management here on the prairies, but I also want to acknowledge Shelley Barkley of Alberta Agriculture and Irrigation and Megan Van Koski of Agriculture and Agri-Food Canada out in Saskatoon as kind of co-authors on this talk, as we put together kind of a bigger session for you today. So there's two main areas that I'm going to explore with you. The first is just talking about insecticides and insecticide resistance and resistance management. And then I want to talk about the insect surveys that are conducted each summer and what happened in 2023 and maybe some things we can expect here in 2024, with primarily a focus down in the Forestburg region, Flagstaff County, but we'll talk about all of Alberta as well. So what are insecticides? Simply, insecticides are really just a substance used to kill insects. They're classified by a committee based off of their mode of action. And the mode of action, and I'm sure most of you are very familiar with this, but it just describes the molecular or cellular target within the insect that is affected by the insecticide, leading to its injury or death. And so there's a variety of different modes of actions. And usually when we talk about mode of action, we can kind of equate that to a group. So we talk about insecticide groups. Each group is those insecticides are put in that group based off of their modes of action. And we'll talk about that right here. So if we think about group 3A, insecticides that are group 3A are all the pyrethroids. Okay, we know pyrethroids act on sodium ion channels. They cause excitation and death of the insects by preventing closure of the sodium ion channels. Group 4As, so these would be the neonicotinoids. And so the neonicotinoids, they all work by irreversibly blocking postsynaptic nicotinic acetylcholine receptors. That is a mouthful. But the reason I wanted to just point out these groups is because often when we're talking about resistance, we talk about resistance in terms of what group of insecticides that insects are resistant to, as well as when we're talking about resistance management, we talk about rotating different groups. And so we rotate between groups because those different groups have different modes of action. So if we think about our insecticides that we have available to us here on the prairies, this is just a snapshot of those available. This is actually the 2022 Blue Book. And apologies for the screenshot of the Blue Book here, but these were those that were listed for oil seeds at the time. And if we look at this list, you probably recognize a lot of these products. But the first off, if we look at the very first one, chlorpyrifos, that's now been banned, removed from the market, no longer available. And then as we go down, I've just kind of listed the groups of where these insecticides fall. So we have group 28, the diamides, that would be Coragen, Coragen Max. We also have the group 1B, the organophosphates like Saigon, active ingredient dimethoate, and so on and so forth. But what I want to just point out here, first off, Matador, as you're all familiar with, the silencer, Matador, active ingredient lambda-cyhalothrin, this now has a restricted use on the prairies. It cannot be applied to any crop that is going to end up with animal feed. So this includes any part of the crop that will end up. So if it's vines or whatever, all of that potential to end up as animal feed can no longer be lambda-cyhalothrin products can no longer be used on those crops. And then same with Volume Express, because that also has the pyrethroid in Volume Express is lambda-cyhalothrin. But if we just were to summarize how many different groups we're not talking very many groups. This is only 4 groups if we went down that list. This is only 4 groups of foliar insecticides, okay? If you look at a few other crops, if you were to look at potatoes or whatnot, sugar beets, they may have a few more products available to them. But if we look at oil seeds, if we look at wheat and barley, these are the main insecticides available to us. So that's not a lot of groups to work with if we want to talk about rotating modes of action as part of an insecticide resistance management plan, especially if we're having to apply a foliar product multiple times throughout the season. Maybe we have to spray in canola for flea beetles. Maybe later on it's lygus bugs. And then who knows, maybe even later on, unfortunately, it's diamondback moth. And so we don't really have a lot of choices in the groups that we're using. And so this is something we really need to be cognizant about. We want to try and use as many different groups as possible, especially when we have to use multiple applications within a growing season. We do have seed treatments. These, again, are crop dependent. So these are the main groups of insecticides you find in seed treatment. So the vast majority are group 4A, the neonicotinoids, but then you can also get the add-ons like the sulfoxamine. So this would be like sulfoxaflor. 4Ds, butanoids, group 28, the diamides again, and group 30, the metadiamides. And so we have a few additional modes of action with seed treatments, but again, there's not a whole lot. So again, something we just need to be cognizant about. All right. And so when we talk about resistance to insecticides, what is resistance? It's really defined as a reduction in the sensitivity of a population to an insecticide when used according to the label. Okay, so we can rule out all other kind of potential mishaps that might have occurred in the product not working appropriately. To be considered true resistance, this really has to be heritable. So the insect has to have the ability to pass this resistance on to its offspring. Okay, so it's linked through genetics. Tolerance, and we talk about tolerance with some of our different insects. This is really the ability of the natural ability of an insect to withstand insecticide exposure, and it's not a result of selection pressure. So if we think about insects are feeding on plants. Plants have natural abilities to defend themselves, different secondary metabolites, different toxins, chemical compounds that they use as defenses. And so insects have evolved alongside plants and have evolved mechanisms or detoxification mechanisms to deal with those plant defenses. And so some of those mechanisms might be co-opted or used or be able to be used by the insect to help detoxify an insecticide. Now, this isn't because we've been overusing that insecticide. It's just the natural ability of that insect to detoxify. This doesn't apply across the board, but it can apply to some of our different insect species. So I just wanted to highlight the difference there between resistance and tolerance. So you're all probably very familiar with how resistance develops. And luckily for us, we don't have a lot of examples of resistance here on the prairies. But basically, if you have a population which mainly has susceptible individuals, as indicated in yellow on this leaf, and you have a few individuals that are resistant, as indicated in red, you apply an insecticide, you kill off, you remove most of the susceptible individuals. Maybe a few survive. They weren't in line, didn't get good coverage, or they're under leaf, whatever. A few individuals survive, but vast majority there's more resistant individuals. They go on to mate. And again, in subsequent generations over time, we apply another insecticide product, same mode of action. We're going to eventually get a buildup of resistant in individuals and a decrease in the susceptible in the yellow individuals in this diagram. And again, so if this keeps happening over more and more time, we can eventually end up with control failure as the vast majority of the populations remain or become resistant to that insecticide. So that's a very kind of simple example of how it could occur. Again, luckily for us, we don't have a lot of examples of resistance here on the prairies. In some ways, we're lucky because we have a shorter growing season, because we have winters, which help to lower our insect populations. We have fewer examples of insecticide resistance here on the prairies. In terms of how resistance actually occurs within the insect, there's a few different ways. And so we call these the mechanisms of insecticide resistance. One is simply target site resistance. And so if we think of this diagram here on the left, we have individuals. An insecticide molecule normally has a specific target within that insect. So whether that's a sodium ion channel or another target, if the insect is susceptible, usually that insecticide molecule can go in to the insect, not fine, but be bound to that target site, and then results in successful death of the insect. But for altered target site resistance is basically the receptor or the location of where that insecticide normally binds is altered. The insecticide can no longer bind to it, so it's not successful. And so the insect can then live, survive, and then successfully pass that trait on to its offspring. We can also have things like metabolic resistance. And this occurs when resistant insects can naturally detoxify or destroy toxins faster than susceptible insects, or can quickly rid their bodies of the toxic molecules so they can excrete it very quickly. So in this, again, this hypothetical example, in a susceptible individual, they might have one copy of a detoxifying enzyme that can work on an insecticide. But in a resistant individuals, maybe there's many different copies or many copies of a detoxifying enzyme. And so that because there's multiple copies, it allows for that insecticide to be detoxified quickly and then prevent it from killing the insect. Other things can occur like physical adaptation. Insects have this, exoskeleton, which has it's a thick cuticle and waxy layer on its surface. And so that can prevent or slow the penetration of some of our contact insecticides, and allowing the insect to be able to detoxify then that insecticide. And again, some of them also might have faster excretion, as kind of mentioned, with metabolic resistance. And finally, we can have behavioral resistance. This doesn't occur that often, but if we think of more things like baits, and even for example, a cockroach going to a bait, they can actually learn to avoid that bait. So they can recognize it and then just avoid it. And so here's, again, just a hypothetical example. Perhaps an insect learns to, be found on the underside of the leaves more often. And so it can avoid the direct contact with that spray. Okay, and these mechanisms, they aren't necessarily all work independently. Unfortunately for us, insects can evolve a variety of mechanisms that will work together to overcome insecticides. Okay, and so there's a variety of factors that can influence the ability of an insect to become resistant. The reproduction rates, generally insects with higher reproduction rates are more prone to develop resistance to insecticides, just because, again, a sheer numbers game, and the chance of one of those individuals having a mutation that leads to insecticide resistance. Number of generations per year. This, again, just allows for a number of individuals to be produced, but also if each of those generations have to be controlled with an insecticide, that's the potential for more selection pressure every single generation. And so the more times we have to use an insecticide, and if it's the same insecticide each year, the more chance there's a potential for insecticide resistance. Migration can work both ways. If we're in a very isolated area and we have insecticide resistance present, then those individuals maybe don't migrate out of that region. But maybe we could have susceptible individuals migrate into that region, mate with the resistant individuals, and maybe we start to see actually a decrease in insecticide resistance. Another kind of worse example of possibility there is where we have insecticide resistance developing in a different area, and then those insects moving or migrating into our region where they subsequently are still resistant to those products. And yes, so that can work kind of two ways. The host range, generally insects with a more narrow host range. They are, for instance, if an insect has, yes, crop species as part of its hosts that it feeds on, but if it also has weed species, the individuals feeding on those weedy species, especially if they're outside the crop borders, they may not be, again, they wouldn't be sprayed with an insecticide, so there's reduced selection pressure on them. So that allows some susceptible individuals to remain. Those with broader host range may unfortunately be exposed to insecticides across a variety of different crops. The persistence of an insecticide or the residual can influence resistance. Usually those that are more persistent in the environment resistance can build to them faster. And then again, this kind of relates to the number of generations reproduction rate, but the number and timing of insecticide applications per year. When we have to spray more insecticides, use them more often with the same mode of actions, that can lead to the potential for insecticide resistance. Okay, so what are some examples here on the prairies? And one that my lab works on is in the alfalfa weevil, Hyperopstica. This is a pest down in southern Alberta, Saskatchewan, Manitoba, particularly in seed production fields. It is also found, of course, in silage and in hay fields, but the resistance issue is really an issue in seed production. And so what we found looking at this is, so we did an experiment where we're testing the adult weevils using commercial grade deltamethrin. So this would be Desis. And looking at, here's the mortality on the y-axis, different doses on the x-axis where the 125 nanograms of active ingredient per centimeter squared. This would be the high rate of insecticide in the crop protection guide. And so as you can see here, so we have the highest rate, then we have half that, so half rate and then a quarter rate. We have 3 sites in this example. Lethbridge, where we have what we believed were naive or susceptible weevils, and then we had 2 sites near Rosemary, Alberta, both where we suspected resistance. And when you look at the mortality, when you didn't expose those insects to insecticide, nobody died, just as expected. Once you use the quarter rate, where we suspected everyone was susceptible or naive to insecticide, we had most of them die, even at the quarter rate. And then as soon as you bumped up to a half rate or the full rate, everybody died. Unfortunately, those 2 rosemary sites where we suspected resistance, we only got to about 50% mortality with even the full rate of insecticide. So that is definitely showing some resistance against the pyrethroids. And we did a survey looking across a chunk of the alfalfa seed production region in southern Alberta. On this map here, it would be those sites with the triangles. And you can see we had just 1 dose. So this is the full rate as well as a control. And you can see even at the full rate, we are only getting about 50% of our individuals collected within these seed production fields dying. So again, indicating that resistance is fairly widespread across these fields in southern Alberta. Another example, this is not from my lab, but that's been tested for a number of years here on the prairies, is Colorado potato beetle. This project was led by Ian Scott and Chandra Moffat, both of Agriculture and Agri-Food Canada. And they published this results just last year. But what they did is they looked at Colorado potato beetle populations across Canada, Alberta, Manitoba, Ontario, Quebec, New Brunswick, PEI, against different products. So those within the neonicotinoids, group 4A, the spinosins, group 5, and diamides, group 28. And then this is a little bit different, but this is basically they're categorizing these populations as whether they're susceptible to those products, they're less susceptible. So they were starting to see, not as successful mortality within those populations. And then finally, those populations that they classified as resistant. And unfortunately, fortunately, Alberta seems to be doing okay. We had a couple of populations that were less susceptible to the spinosins, and only 1 population that was less susceptible to the neonicotinoids. Unfortunately, Manitoba not doing so great, with many populations tested less susceptible or resistant to a variety of products that were being tested. Another example would be the soybean aphid. This is not known. It's not known if they can overwinter on the prairies. It's thought that they really can't. And so it's one that kind of similar to diamondback moth. It's blown up on wind currents each year. And John Gavlosky with Manitoba Agriculture and colleagues down in the US did a study a few years ago looking at a couple of different products, both group 3A, lambda-cyhalothrin, which unfortunately, again, is no longer available to us now, but at the time it still was, and biphenethrin, which is not available here in Canada, but it is in the United States. And so just to highlight those Manitoba populations, there's 3 Manitoba populations. This time is proportion mortality on the x-axis. And so we can see a laboratory check. It had basically almost 100% mortality. And then you can see decreasing mortality across these populations, and including those found in Manitoba, 3 different sites in Manitoba. And for both products, resistance was noted. And so this would be, again, one of those where migration is kind of a culprit, or is leading to us having resistant populations of insects here on the prairies. So one that's often talked about are striped flea beetles and the difference in their susceptibility to the neonicotinoids. Some work led by James Tansey when he was a postdoctoral fellow here at the University of Alberta, as well as the late Lloyd Dosdall and Andy Ketty did some work looking at striped flea beetles as well as crucifer flea beetles. So crucifer here on the left, striped on the right. And what they noted is there was just reduced mortality to the neonicotinoids during that spring generation in the striped. They did not note that in the crucifer. And so this has been kind of talked about for a number of years now, and it seems to still be the case where the neonicotinoids do a better job at controlling the crucifer rather than the striped. And it's thought, again, that this is tolerance. It's thought that it developed, or it was already kind of present within the natural ability of these beetles to detoxify just chemicals. And so they had a better ability naturally to detoxify the neonicotinoids rather than resulting from selection pressure from the neonics themselves. Something that we need to be aware of, I guess, is the diamondback moth, so Plutella xylostella. So this moth, again, we know doesn't overwinter here on the prairies. Each year it's blown up on winds from the south, and our friends just directly to the south in North Dakota have found issues with control with pyrethroids. So this is noted from their crop and pest report in late August 2021. And they did confirm resistance in these diamondback moth. So this is something we need to be aware of too. There are populations down in the southern United States, especially where a lot of brassica vegetables are grown, where a bunch of insecticide products are no longer available, or like no longer control diamondback moth. And so this is something we do need to be wary of here on the prairies, is that there is the potential with these diamondback moth populations being blown in. They may have resistance already built in and within them. And so something to be wary of. If you spray a synthetic pyrethroid and you don't get good control, then rotate out of that and use a different product that might have resistance to it. Cabbage seed pod weevil. This is not one that has resistance here in Canada, but it's something, again, we need to be aware of. There has been resistance noted to the pyrethroids in Germany as long ago as 2011. And just to show you on this map, both these sites labeled with the number 3, that's where resistance was first noted. And so invariably, each summer, fields are sprayed for cabbage seed pod weevil here in Alberta. And so it's just something we need to be wary of and on the watch out for. So what can we do? How can we avoid putting enough selection pressure on these insects to develop insecticide resistance? And a major one is, avoided repeated use of insecticides from the same chemical group or subgroup. So like, don't just keep using pyrethroids, especially multiple times during the year on the same crop. Might be for targeting different insects, but those different species some of them still might be lingering, and so they might be exposed to several applications. So avoid that. Where possible, use some of the more selective insecticides, and of course, good integrated pest management strategies to preserve natural enemies and let those natural enemies help to manage some of our insect populations. Use products at their full recommended rate. It seems pretty obvious. If you can, target younger larval instars because they're usually much more effectively controlled than the adults. This is particularly true with grasshoppers. And of course, grasshoppers also, when they're bigger, they eat more. So if you can control them when they're younger, you should get them before they cause significant damage within the field. There's economic thresholds available for a variety of our pest species. So use economic thresholds and appropriate spray intervals. And yeah, I like to say a lot of our best management practices are already uh, the foundation for great integrated pest management. And so keep that up and then use these other strategies as well. Help our beneficial insects if we can, rotate our insecticides, use some of our softer chemicals and more selective chemicals when we need to. All right. Okay, so now I'm gonna, and yeah, so I guess overall, I think really with thinking about resistance and resistance management plan, I think if we get a plan in place, we can really avoid insecticide resistance here on the prairies for years to come. Okay, now I want to switch gears a little bit, um, and I want to talk about insect pest surveillance here in Alberta, but also across the prairies. So here in Alberta, there's an Alberta Insect Pest Monitoring Network, which hopefully most of you are familiar with. It's currently being led by Shelley Barkley, a technician with Alberta Agriculture and Irrigation. And as part of the Alberta Insect Pest Monitoring Network, they feed data into the prairie-wide Prairie Pest Monitoring Network. And the Prairie Pest Monitoring Network is headed by Megan Van Koski at Agriculture and Agri-Food Canada and Jennifer Otani up in Agriculture and Agri-Food Canada and Beaver Lodge here in Alberta. So each year they conduct surveys along with help from many of you, probably even online. And they look at a few different insect pests. One is the pea leaf weevil. It is monitored each spring, usually I think before the around the 3-node stage. And then the notches on the clam leaf are counted. And then compiled into a nice map. Here in Alberta, there's 215 fields that were surveyed last summer. Some areas where we do see high levels of pea leaf weevil feeding damage, there might be maybe warranted to think about a seed treatment on your pulse crops there. So just to show you the maps, this is what the 2022 survey looked like. With the darker colors meaning higher levels of damage. Basically, anything over 27 notches per plant would be economic. And so here is the 2023 survey, and you can see particularly northeast of Edmonton, well, surrounding Edmonton, north and east, and down into Flagstaff County, there is quite high levels of feeding that was noted in this spring. And historically, this was considered kind of a southern Alberta pest below Highway 1 between kind of Calgary and Medicine Hat. You can see that's no longer the case. It's well established up here in our region, and it's something we should be thinking about a management strategy for going into this coming field season. For cabbage seed pod weevil, 220 fields were swept in Alberta, all the way from the U.S. border to Athabasca County. It should be noted that Jennifer Otani does conduct a canola flower a survey during canola flowering up in the Peace, and no cabbage seed pod weevil were noted up there. And so they are collected, and then the insects are sorted, and cabbage seed pod weevil counted, and again a survey map produced. And so this map is the 2022 map. And then if we go over to the 2023 map, you can see a dramatic increase in summer 2023, most likely probably due to some of our growing conditions, really that hot, dry weather. These insects can do quite well in. And so this is definitely something that should be on our radar for this upcoming growing season. And I want to point it out because even though on this map, it's not in Flagstaff County or even in Beaver County. It has been noted even further north of Edmonton. So there is potential for range expansion. And in previous years, this range has been found further north here in Alberta. So it's just something to be aware of in our fields. Wheat midge, 253 fields sampled. Um, and there's just some extra information here. So there's some areas that weren't sampled, some of the special areas, only irrigated fields down south. Um, and then, yeah, anyways. So basically, the midge, this is a fall survey using soil cores. Soil cores are taken in the fields, brought into the lab. Shelley then looks at the cocoons that are in that soil and determines whether or not those larvae overwintering in those cocoons are viable, and a map is produced. Midge do really require spring rain to complete larval development, pupate, and then emerge as adults. So if we don't get good rain in May and June, we may not get a flush or a large flush of midge emerging. And unfortunately, they have been shown to be able to survive up to 11 years in soil within their cocoons. So here again, here's the 2022 map with kind of a hot spot here. I guess this is Camrose, Flagstaff, and Beaver County. And then from the 2023 survey, a little bit more spotty. Some other little hot spots have popped up. But I like to think of this as kind of a snapshot. Just if you're in one of these light green areas, doesn't mean there's going to be no issues with midge. We do want to keep monitoring for them. Again, this is Shelley does a great job to get out to several fields, but often it's only about 3 or 4 per county. And so what's happening in your field might not be the same as your neighbor fields or those fields that were surveyed. So it's just something, if you're in one of these hotspot areas, definitely pay attention for midge this coming year. And if you're outside that, we should still be on the lookout for midge, especially historically if you've had midge issues in the past. Okay, wheat stem sawfly, generally considered southern pest. So we have seen increasing population densities, particularly because of the drought conditions over the past few years. There's little in-season, there's basically no in-season management strategies available. Prior to the growing season, you can grow a solid stem variety, you can rotate away from wheat. During the growing season, if you do notice that if you are monitoring for them and see high levels of high population levels, you can potentially swath to prevent the lodging. It's you're controlling the lodging then. If you harvest higher up, so leave more stubble, that allows for, there's a wheat stem sawfly parasitoid, which also overwinters in the stubble. So by leaving higher stubble, we can allow the parasitoid to survive and emerge as well. So last year there was 87 fields in 20 municipalities in Alberta. Here's the 2022 survey. So some pretty high numbers, especially further down south towards the border. And then in 2023, we did see an increase in density and area affected. Okay, so and it is creeping up, and in Saskatchewan, it has been found fairly far north as well. And so it's something to definitely be aware of and be on the lookout for. Probably 2023 was the year of the grasshopper. Everyone was talking about grasshoppers. Where they were saying, where did the flea beetles go, and where did the grasshoppers come from? So a huge grasshopper year last year. Development was about 2 weeks ahead of schedule last year. So this is pretty significant. It got some really large populations when the crop was still, doing a lot of it still had a lot to grow. So very high populations, especially in southern Alberta and southern Saskatchewan. Megan took this photo of one of her summer students. Taylor counted over 1,700 grasshoppers in 25 sweep samples taken from a ditch near Kindersley, Saskatchewan. So extremely high population levels. So there's a survey done each year for grasshoppers. Here's the 2022 survey. And then here's the 2023 survey. And you can see kind of a bit more widespread, but also the increased intensity in the numbers of grasshoppers we have. Up along the parkland into the right along the boreal and up in the Peace region, we have this Bruner's grasshopper, which we seem to have a 2-year life cycle. So we see the odd years, you generally see higher levels of grasshoppers up in the Peace and in the parkland. And so this means that potentially in 2024 in the Peace region along the parkland, we might see less of a grasshopper issue. But further south, I think, especially given our, I guess, very warm winter, at least until January, that probably allowed a longer oviposition period into the fall. And so we probably can expect some significant grasshopper numbers this coming year, unfortunately, and especially if this kind of hot, drier weather continues. Okay, cutworms is something that is not actively surveyed for by the Prairie Pest Monitoring Network, but there is a reporting tool. And this reporting tools basically just kind of indicates where some hot spots or some fields that had some cutworm damage. Cutworms are very patchy and difficult to predict across the whole province. Yeah, they can be active when it's cool. And so, yeah, sometimes we don't expect there to be feeding, but unfortunately they're there, they're feeding. Again, similar to grasshoppers, if we have a long, prolonged fall, nice warm weather, it gives a lot of the female cutworm moths more time to oviposit. Also, if we kind of think about it another way, if we do have early harvest and we take off our crop earlier than usual, sometimes we get some regrowth or some weeds growing. The cutworm adults like those and aim for those spots as well to lay eggs. And so it really leads to this very patchy distribution and something, unfortunately, that we're not very good at predicting when we might see outbreaks in the field. There is a reporting tool. So if you have your smartphone on you and you want to report cutworm damage into this coming growing season, you can scan this QR code and you can report there. Okay. And you can just see some more information about that online. Okay. With that, I just want to thank you all for attending. I want to thank all the funders for my research program who helped make our research happen, especially the commissions here in Alberta who support my industrial research chair along with NSERC, but as well as, of course, WGRF, ARDAR, etc. Everybody on here gives us funding to help us develop and continue to monitor insect pests here on the prairies. All right, with that, I'd be happy to take any questions there may be. Oh, I can't hear you. Oh, there.
Oh, can you hear me now?
Yeah.
Yeah. Thank you so much for this amazing presentation. Now, if any participant have any questions, feel free to write in the comment section or in the Q&A.
I know I didn't talk about flea beetles, but that's because flea beetles kind of dropped off last summer. And so it's kind of one of those where did they go was kind of the main question I got about flea beetles last year. And it just shows their unpredictability of them, unfortunately.
Yeah, let's wait for 5 minutes. If there is any questions, we can answer that. If not, and then we can end the webinar. Are there any questions?
All good if there's no questions. That's fine. Hopefully, you might have heard some of this
Yeah, I
information
guess.
before. So,
Yeah, I guess there are no questions.
yeah.
Okay.
Oh,
Yeah.
all right.
Thank you
Cool.
so much for. Hello. Can you hear me?
Yep.
Yep. Perfect. Yeah. I think so. There's no question at all. Yeah. Thank you so much for giving this excellent presentation. It was truly a pleasure to have you here with us today. And to all the attendees, we appreciate your participation in today's webinar. We hope you found this presentation informative and enjoyable. This marks the end of our session. Thank you again for joining us. If you have any further inquiries or feedback, feel free to reach out to us. Have a wonderful day. Yeah. Thank you so much again. Yeah.
All right. Thank you.
Yeah.
Take care.
Yeah. Thank you.
All right.